for coming to our last Science Under Stars event of this semester. My name is Tianyi, and I'm currently a third-year PhD candidate at UT Austin, and I'm one of the co-presidents for Science Under Stars. And today we have Wenli, who is currently a postdoc at the Department of Molecular Biosciences at UT Austin. She graduated from the plant biology PhD program from UT Austin last year. So she'll be talking about how plants change our world today. So let's welcome Wenli.
Thanks for inviting me today. I'm really happy to have this opportunity to talk with everyone in this cold weather while it's warm here. My name is Wenli. I'm a postdoc in a plant biology lab in UT Austin. So today I will talk about plant genes and what we can do with plant genes. So I assume most of you know what is gene. Gene is DNA sequence that carry genetic information. So different species, they have different genes. That's why we look so different. In the lab, we use a plant material called Arabidopsis. It's a kind of grass. It's a model plant. They look like this, very happy in the chamber. Before I joined to UT, I was working on another plant called Brassica rapa. So it's a kind of vegetables, and it's very common. It's very healthy for your body. So when I was a graduate student in Nanjing Agricultural University in China, we had a huge field, and we need to do a lot of work in the field, especially during the winter season. It's the harvesting season for this Brassica rapa. And this is my previous professor. He was checking those cabbage. Brassica rapa is just the academic name. It's also called cabbage or Chinese cabbage. In HEB, I noticed they were labeled as Napa cabbage or bok choy. So they're just same thing. The Brassica rapa is a 2-year plant. So you have to plant them usually during the spring, and they will grow big, and you harvest them during the winter. Once they start flowering in the next year, spring, it can grow very high. The relationship between Brassica rapa and Arabidopsis is very close. They all belong to Brassica family, but although they look very different, one is huge, a vegetables, but the Arabidopsis is very small plant. You probably can find them in the wild. They are tiny plants on the ground with little white flowers. But in the lab, we always give them the best conditions to grow. So they need to be very healthy for our experiments. So why we need to study on plants? Because plants are very important to us and to our ecosystem. But different from animals, plants cannot move around freely. In the cold weather, we can wear more clothes, we can stay in the building, but for plants, they just have to stay where they grow and deal with it. However, like us, plants also watch the sunrise and experience the changing seasons and breathe the air in different ways. So I will tell you how. So first, plants can actually sense or see the light. If you had any experience growing sprouts at home, sometimes you might see those seedlings, they bend to the light, especially when the light comes from one side. That's called phototropism of plants. So plants can actually track the light. And if we put the seedlings in the dark condition, they will grow very weak but longer because they try to grow faster to reach the light. Once the plants grow in the light condition, they look very different. They look healthier and stronger, but they grow shorter. They have the open and wider leaves to receive the sunlight. So those are some evidence that plants can sense the light condition. Then what about temperature? Different temperatures have different effect to plants. And adaptation to the changing season is very important for plants. Different plants have different temperature needs. Imagine if some plants, they flower or produce the seeds in the wrong season, they will have problems. So some plants they need cold temperature, but some plants need the warm temperature. Here, this is a very famous blue bonnet in Texas. You can find a lot of them in the wild, and most of the plants, they flower in the spring because spring has appropriate temperature. But some plants, for example, winter wheat, they need the cold temperature. So in the north area, you have to plant winter wheat before the winter. Usually it's in the autumn season. And after the winter, they accumulate enough cold temperature, they can flower and produce wheat. Some plants can even flower in the winter. Here, this is the plum blossom. They can flower in the winter even when it is snowing. So plants are very different. So here, I'm going to show you a very interesting and important plant organ called stomata. Anyone of you heard stomata before? Good, some of you at least. So stomata is a little pore on the surface of the leaf. It's formed by two jelly bean-shaped gut cells. So those are two gut cells. When the shape of the gut cells change, stomata can close and open. And plants use stomata to exchange the gas and water. They can take the carbon dioxide from the air and they release the oxygen. That's why plants are so important for us. So we breathe with our mouths, and plants can actually breathe with their stomata. So you can see stomata as plant mouths. And stomata is very small. It's difficult for us to see with our eyes. We have to use microscope. Those are stomata under the microscope. You can see these are two gut cells, and stomata can open and close. And those green dots in the gut cells, they are chloroplasts. That's why plants show green. And in the lab, we use a different way to observe the stomata. We use a kind of staining. So you stain the plant tissue or the plant leaves, and you put them under the microscope. The staining will stay on the surface of plant cells, so you can see clearly the shape of plant cells. We can see here, those are mature and open stomatas, and some stomata are in the process of development. And all the other cells, they are pavement cells. You can even use some markers. For example, here I use this marker called nuclear GFP. They stay in the nucleus of the gut cells. So you can see clearly where is the gut cells. There's some gut cells with the nucleus, so they are stomata. Very interesting. So now I've shown you how plants sense the light and how plants experience the temperature change and how plants breathe with their mouths, stomata. The question we want to ask in the lab is, what is controlling all these plant behaviors and plant responses? And who is controlling the plant whole life cycle? Because plants are so different. They have so many different behaviors and responses according to the environmental change. And the answer is genes. So as I just mentioned, gene is DNA sequence that located in the nucleus. If we enlarge the plant tissue or plant leaves, you can see a lot of cells, and each plant cell has one nucleus. And each nucleus, they will have those different DNA sequence or genes. And those genes in the nucleus will finally be translated into different proteins. So plants that will affect plants' behaviors and response. For example, during the cold weather, those cold responsive genes will be activated. So they will produce a lot of cold responsive proteins, which will help plants to cope with the cold weather. So gene is the conductor of life, including both plants and animals. Here I'll give you some examples how scientists find different genes control different responses in plants. First, we know that plants can actually sense the light. So scientists found a gene called 5B. When they knock out this gene, that means when they silence this gene or delete this gene in the plant, it's like blocking the plant's eyes. they cannot sense the light anymore. So they thought they were still in the dark condition. They're trying to grow faster and longer to reach the light. So except for 5B, there were a few other photoreceptors in plants were identified. So scientists found actually the plants can sense the light in a much larger range than us. From far red light to UV light, use different photoreceptors. For example, they use UVR8 to sense the UV light, and they use 5B to sense red light, 5A to sense far red light. So those photoreceptors can be seen in plants' eyes. So instead of 2 eyes, plants have a lot of eyes. And then they can, according to the light condition they are growing, they can make different responses and behaviors. Then what about flowering? We know that temperature can affect flowering behavior in plants. And scientists find a gene called FT. When they knock out this gene, or when they silence this gene in the plant, this plant cannot flower, or the flowering time will be significantly delayed. But when they overexpress FT gene in this plant will flower much earlier. So FT gene can promote flowering in plants. Another gene called FRI, fry. In fry lines, these seedlings just keep growing leaves. It will never flower, or sometimes some of them might flower, but flowering time is significantly delayed. So imagine if we have some vegetables carry fry genes, so you just keep growing leaves and we have endless vegetables. So here, this is flowering promoting pathway. We don't have to memorize this, I just put it here. So flowering behavior in plants can actually controlled by multiple genes. And these signaling pathways took scientists decades to figure out. Here, let's come back to the stomata again. As we know, plants breathe with stomata. They use the carbon dioxide from the air and release the oxygen. So usually, stomata also can close and open according to the condition. In hot summer day, those stomata will be open. So that time, it's not a great time to water them because stomata open, and if you water them, the water evaporation will be increased. So those plants might be wilting. So the better watering time is early morning or later afternoon. But we want to figure out in the lab is what genes behind the stomata development. So there are some very interesting genes. This is a wild type plants or a control plants. We can see in the wild type, they have functional stomatas. But scientists found if they knock out the gene, or if they silence the gene, then this plant will develop into this no stomata phenotype. They all pavement cells, no stomata. So this plant cannot breathe. It will just die. And this gene was named as speechless. And followingly, another gene was also identified. And people found if they mutate this gene, this will change this protein's stability. And this plant will develop into this all stomata phenotype. So every cell, instead of like this, every cell becomes stomata, this crazy looking. And this gene was named as scream. So currently I'm working on both speechless and scream genes. Usually who first identified this gene, and then they got the chance to name this gene, which is really cool. Except for speechless and scream, there are a few other genes were also involved in stomata development. They are mute gene and pharma gene. So when we knock out mute gene, also this plant doesn't have functional stomata, they have those small cells. So that's why this gene is named as mute. And the pharma gene, if we knock out pharma gene or silence pharma gene, this plant can also not develop those mature functional gut cells and the stomata. So they form these multiple layers of cells, look like a hamburger. So the stomata development needs all of those genes get involved. They have to collaborate with each other. If any one of them has problems, then the stomata development will stop there, and plants cannot have functional stomatas. So those are some genes functions, and the gene already being identified so far. This is what we do every day in the lab. We identify genes function and try to figure out the gene network behind the different plant behaviors and plant organ development. And once we know a gene's function, we can upload or publish the paper, so those genes function will be collected in the database. So you can actually search what gene has what functions. And if it's very useful, it can be used in a lot of fields and aspects. For example, one of the most common technology used right now called transgene. So you generate transgenic plants. It's also a way to confirm a gene's function. Here is the simplified process of generate transgenic plants. So we use a bacteria called agrobacteria. In the early days, scientists found agrobacteria has this ability. They can transfer their own genes into the plant by infecting plants. So these plants can carry the gene from the agrobacteria. Then scientists was thinking, oh, maybe we can use this ability. What if we put a gene that we are interested into the agrobacteria? And then we infect this plant so we can get the transgenic plants, which carry the gene that we are interested in, and usually through tissue culture. So here we can cut off the plant tissues, usually plant leaves or roots, into small pieces. Then you put them in this media, which contain the agrobacteria. And the agrobacteria can carry the gene that you are interested. Then they will grow into this whole plant in the container. And you can test if this plant is carrying the gene that you are interested in. If they are, then you can transform them into the soil, let them grow, so you got the transgenic plants. So does this mean we find the key to engineer life? We can transfer a gene from one plant to another plant to give the transgenic plants a new trait. Imagine if we transfer some gene from orange into the apple, and then we get this orange apple. It looks like apple, but it tastes like orange. Or maybe we can transfer some genes from watermelon to pineapple, and we can get this new fruit. Looks like pineapple, but it tastes like watermelon. In the early days, scientists were also thinking, what if we merge a cell from tomato to a cell, potato cell? So if we merge these two cells, that means we combine these two different genomes. Maybe we can generate a new plant which produce tomatoes overgrown and potatoes underground, so we can get double yield. And they did get this plant, but they didn't get the tomato and potatoes as expected. So plants themselves also have the mechanism to resist any changes from outside. But we did get some successful transgenic plants so far, which is available on the market. So I'll give 3 examples, which is very common, but I don't know if everyone know actually we are using them. So first is called BT cotton. Cotton is very commonly and widely grown crop, but there's a worm called bollworm. They live on the cotton and they eat the cotton leaves or any part of the cotton. So this usually causes huge economic loss. And then scientists found that's a kind of protein called BT protein, which is toxic to this bollworm. So they transfer this BT gene from bacteria. They found this gene in this bacteria into the cotton plant. Then they got the transgenic cotton and then named them as BT cotton. So if the worm live on the cotton and eat any parts of the cotton, they couldn't digest this protein and they will just die. So by this way, people can get more yield of the cotton and higher quality of the cotton. And the same technology can also be used in corn. So far, 90% of cotton acre were planted with Bt cotton. Another example is golden rice, which is not commercially available right now, but it was developed in the 90s to address the vitamin A deficiency in children and women. So scientists transfer a vitamin A gene from another plant to the rice. So the rice can produce this rice with high level of vitamin A, which give the rice this yellow and golden color. So this significantly increased the nutrition of this rice. The other example is papaya. I'm not sure if everyone knows, actually on the market right now, over 80% of the papaya, especially Hawaii papaya, they are transgenic papaya. So in papaya, there's a virus called the ring spot virus. They can infect the papaya plants, which usually will kill the plant and the fruit. They will look like this. So scientists transfer 2 genes from ring spot virus into the papaya plants, and the transgenic papaya plants will have the resistance to this kind of virus, but doesn't affect papaya plants' growth. So this technology saved the Hawaii papaya. So transgenic or transgene technology can be used for plants. It can also be used for animals, but not just for food. But every time when those transgenic plants become our food, people become concerned or worried. And of course, we need to be concerned, especially as a scientist. So a lot of people talk about GMO. GMO means gene modified or gene edited or sometimes bioengineered organisms. It includes both plants and animals. And all those examples that I just mentioned, they are all GMO. So there was some concern about GMO food. Currently, those available GMO food on the market is not a lot. Here is just the top 8 GMO crops in the US. They are cotton, sugar beet, soybean, corn, canola, papaya, alfalfa, and summer squash. So most common one is cotton, soybean, and corn. A lot of them either have the resistance for insects or herbicides or different virus and stresses. So they can, by using these GM crops, it can significantly decrease the use of herbicide and increase the quality of these products. And of course, there are also a lot of debates about GMO food. Even though GMO crops can increase the quality and decrease the use of herbicide, which is good for our environment, but also there are some concerns. Even my parents would ask me, are they safe? Are they cause cancer or something? So they don't directly cause cancer, but there are some environmental issues or ethical issues. So it might happen during the testing if a gene that we transferred, if it escapes into the wild, that might result in a supergrass, which can be out of control, that's called gene escape. Also, if some people transfer a gene from an animal into a plant to increase either the quality or the nutrition of these plant products, but that plant can be considered as vegan, so that's debatable. But as a customers or scientists, we need to know how the transgenic plants were generated so we can make our own choice. The transgenic crops can be used in a lot of other ways. They can produce environment-friendly power or energies. We can also use transgenic plants to produce some enzymes or proteins which is difficult to get from animals or other technologies. This will benefit our medical system. And recently, there's a company called Lake Bio. They generate this new plant called glowing plants. They transfer a gene from a firefly into the plant, so this plant can glow at night. Imagine we have those plants in the yard or in the park, so we don't need electricity anymore. And they're actually commercially available right now. You can get them with $30. But I haven't seen them in Texas yet. So this is the last part of my talk today, and this is the take-home homework. So next time if you go to groceries, you can try to find some GMO products. Usually they will be labeled as genetic engineered or bioengineered, and so far there are only 10 GMO crops available in US, and same as that in other countries and areas. So you can see if those products are made by any of those. The most common would be potatoes, soybeans, and corn. And also, a lot of food you will find them, they label as non-GMO or organic. So, yeah, see if you can find them in the grocery and just check the package. And yeah, thanks for being here today. And if you have any questions, feel free to ask me. I actually have two lab members here today, Calvin and Answer. They were also working on those projects with me. So feel free to talk with them. I'll take any questions.
Didn't I understand you to say that the plant had a gene from a firefly that had bioluminescence?
A gene from,
I thought you said a firefly.
yeah, firefly.
So does it have, is it bioluminescent? The plant?
Yes, it is.
Yes, that.
It is. It's the same mechanism as like in the firefly. So that's why the plant can glow.
Wow.
Yeah. And those are just some pictures from AI or like Avatar, yes. Would any one of you like willing to consume the GMO food or try to avoid it?
I'm curious, do they have to be labeled? You talked about finding in the grocery store.
They
I've
have
never
to be
seen
labeled.
labels.
Yeah, they have to be labeled.
They have to be labeled.
Yes. So every
And what about
GMO,
I mean, produce doesn't have labels on it. Is that like on the box? Is that all it has to be?
it should be on the box. If they use any ingredients from GMO food, they should be labeled.
So like summer squash, and you go and there's a bin of squash, it's not labeled.
If the raw squash, maybe it's difficult to find out, but from the whole package you might find out, yeah. So each GMO food, before they available on the market, they have to go through a series of testing and field testing. I think that's why the golden rice is not available right now, because when people are doing the testing, it's a lot of debates. Things should be done that way or something, because they have tests with humans. Yeah.
What's your favorite genetically modified plant?
That's a good question. Actually, in the lab, we did a lot of we did transgenic plants every day, but that's just to identify or confirm the gene's function. It's not, we don't produce them in the market, we only use the mono plants. But just considering those available GMO plants, I think it's baby cotton. I think that's genius who found this and get the baby cotton very stable to grow. It saved the environment. Yep.
Do you yourself eat, like if you had a choice between, let's say, 2 apples, would it matter to you?
I do eat. I don't mind eat GMO food once it's properly passed the testing and everything. But in the grocery, I also tend to buy food which is labeled organic. But that is just more try to avoid the additive, the artificial additive, because except for GMO, currently I think a lot of big companies, they tend to use different chemicals, some additives to make the food taste better. But yeah, it's more the additive thing, not the GMO thing.
So thank you for a great talk. That's really informative. I'm curious, you've mentioned these 10 different plants that are available as GMO, and
On the market.
they're ostensibly, there's no harm that comes from those. Were there any experimental plants along the way that were attempted that actually did cause any harm or had any reason to not be allowed on the market?
As I know, just considering about the nutrition, I don't think that caused any obvious or direct harm to any animals, but it does have some environmental issues. So like the BT gene, because sometimes people find if in the wild, sometimes the crop can also get accidentally crossed with other wild plants. So if they get crossed, the pollen, the pollination happened, the BT gene can get transferred into the wild. So that's something scientists don't want to see, because that's called gene escape. If they into the wild, something out of control, that might not be bad. That might not be good for the environment. So I think that's the concern. I think Monsanto have some GMO crops. They do the field testing. So they have to label this whole field, and then alter this field certain miles. They have to check that miles, and no other crops grow there. Yeah.
I can add one comment to the question. So I think for the scientists, the biggest concern maybe is the loss of genetic diversity. So these kind of transgenic plants, they are going through the same batch. They don't have too much genetic diversity. That's why there is no other health or bad health effects for the genome crops or the fruits. So it's better, maybe better than the plants or fruit which are heavily insecticides or pesticides. Better to use the GMOs better than those which are sprayed heavily by the insecticide. So that's why they built this BT cotton, because previously they were controlling this worm through the insecticide. So after they have this from the bacteria transform this gene, the crop is naturally resistant. So you don't need to spray the insecticide. It saves billions of dollars, and also the crop is healthy under this transgenic plant.
Any questions from the students? I think I just started learning about GM technology when I was in high school. Okay.
Thanks, everyone.
Thank you, everyone.
We have a very exciting program signed up for next semester. Our first talk is going to be on January 16th, and it's going to be about leaf cutter ants. And then the second one is on February 22nd, and it's going to be about bumblebee. And that one is partnered up with the Texas Science Festival, so it's going to be a big event. All right.
Okay.
Cutter ants study here.
Huh?
Cutter ants study here.
So you have your
Yeah, we're just at UT.
it's studying at UT in general. That speaker is